let's start our protein subunit presentation. Our group member consists of Ms. Siti Haya, Nurain Masturina, and Siti Aisyah. The introduction of protein subunit. Protein-based subunit vaccines present an antigen to the immune system without viral particles using a specific isolated protein of the pathogen. And now, how does the vaccine works? The only one that uses the most basic sections, the subunit of a virus or bacterium that the immune system has to recognize, does not use a healthy virus as a vector or hold the whole microbe. Subunit vaccines contain pathogen proteins and all polysaccharide fragments, probably researched to determine which variations of these molecules are likely to elicit a robust and efficient immune response. The risk of side effects is reduced by limiting the immune system access to the pathogen in this manner. For example, against COVID-19, the protein subunit vaccines contain harmless fragments proteins of the virus that induce COVID-19 rather than the whole germ. Our bodies know that the protein may not be present and produce T lymphocytes and antibodies that can recall how to combat the virus that induces COVID-19 if we get sick again. Examples of vaccine developer: Marius Rob Helleman, Baruch Samuel Blumberg, Pablo Valenzuela, Leila Alice Demark, Richard Shalagan. For hepatitis B vaccines, there are three inventors. First, Marius Rob Helleman, a leading American microbiologist who specialized in vaccinology and developed over 40 vaccines including hepatitis B, an apparently record of productivity. Second inventor, Baruch Samuel Blumberg, American physician, geneticist, and co-recipient of the 1976 Nobel Prize in Physiology or Medicine for his work on the hepatitis B virus while an investigator at the NIH. Next inventor, Pablo Valenzuela, a Chilean biochemist dedicated to biotechnology development. He is known for his genetic studies of hepatitis viruses, participated as R&D director in the discovery of hepatitis C virus and the invention of the world's first recombinant vaccine. This is hepatitis B vaccine information. First, contain hepatitis B virus surface antigen HBsAg derived from the hepatitis B virus, was the first subunit vaccines to be accepted for use in humans in the United States. Second, the initial hepatitis B subunit vaccine received HBsAg from contaminated individuals' plasma. Modern recombinant technology has provided for the development of these vaccines without the use of human plasma samples. For acellular pertussis epi vaccine, the inventor is Leila Alice Demark, an American pediatrician in Atlanta, Georgia, the one that first discovered pertussis vaccines on freeze-inactivated Bordetella pertussis bacteria, introduced in the 1940s century. This is acellular pertussis epi vaccine information. First, another kind of subunit vaccine made up of inactivated pertussis toxin and one or more other components of this bacteria microorganism. Second, the pertussis toxin is detoxified either by treatment with a chemical or by using molecular genetic techniques. For human papillomavirus HPV vaccine, the inventor is Richard Shalagan, an American scientist and professor. He was the chair of the Department of Pathology at Georgetown University from 2000 until 2019 and is now the director of the Center for Cell Reprogramming at Georgetown. This is human papillomavirus HPV vaccine information. First, consists of recombinant viral vaccine proteins containing highly purified virus-like particles (VLP), which are protein shells of the HPV virus. Second, virus-like particles (VLP) contain no viral DNA, thus they cannot infect cells, reproduce, or cause disease.
Vaccine trials conducted. Clinical trials can divide into three phases: Phase 1 vaccine trials, Phase 2 vaccine trials, and Phase 3 vaccine trials. Throughout all phases of clinical trials, the safety of a vaccine is the main priority. Early phase trials examine whether the vaccine induces the development of antiviral antibodies, while late phase trials evaluate whether the vaccine can prevent people from getting sick.
Phase 1 vaccine trials. This attempt involves a small group of adults between 10 to 100 healthy volunteers. The volunteers will receive different doses of vaccine. The common adverse events occurs include redness at the injection site, muscle pains, headache, and fever. The researchers will first test adults and then gradually step down the age of the test onto the child. The Phase 1 vaccine trials may be open label in that the researchers and perhaps volunteers know whether a vaccine or a placebo is being used. Phase 2 vaccine trials. It's same as the Phase 1 vaccine trials, but it focuses on adverse events and immune response. This testing involved a large group of several hundred individuals participated. The several individuals may be in a population that is at risk for infection of the disease. The best way to assess vaccine effectiveness is to measure antibody levels. To study the Phase 2 vaccine trial is to study the candidate vaccine's efficacy, immunogenicity, suggested doses, immunization plan, and method of delivery. The phase three vaccine trial is largest trials which involving thousands to tens of thousands of people. The tests are randomized and double-blind and involve the experimental vaccine being tested against a placebo. A certain uncommon side effects could not have shown in a smaller group of subjects tested in early phases, but this will shown in the phase 3 vaccine trials. The phase 3 vaccine trials goal is to assess vaccine safety in a large group of people. A vaccine may be listened for use after a successful phase 3 trials.
Vaccine efficacy. There are various factors must be controlled to verify vaccine effectiveness. First, levels of antigen-specific antibodies. Second, the immunization dosage. Third, adverse effects. And the number 4, the type of immune cells reacting. Many problems must be overcome and many research and development working together in order to make sure that the vaccine is safe before trades it on market. A vaccine candidate is selected based on the antigen's ability to induce defensive immune response. Pre-clinical trial will begin after research produces a vaccine candidate. Before clinical tests started, pharmacological and toxicological risks of vaccine candidates should be evaluated. This is because to make sure that vaccine is safe and effective. Advantages: protein subunit vaccine is safety because the components only contain recombinant proteins or synthetic peptides without the involvement of infectious viruses. Number 2, vaccines made up of protein subunits seldomly cause side effects at the infection site. Number 3, an ideal subunit vaccine must be safe, stable, and immunogenic in all immunized individuals. These features have made subunit vaccines an attractive vaccine candidate. The manufacture's purification and characterization should be efficient and be inexpensive in order to enable the widespread vaccine use.
For this slide is about the advantages. Subunit vaccines do not contain live component of the virus but are made from purified pieces of the virus, which is protein antigen that trigger an immune response. This is an existing technology used, for instance, in hepatitis B vaccine, but it has several disadvantages, which are the protein antigen that the best elicit an immune response must be investigated in detail. Also, it can stimulate an insufficient immune response, meaning that protection is likely to require multiple booster or for the vaccine to be given with an immune system stimulant. So next is about the side effect to human post-vaccination effects. Getting the COVID-19 vaccine will help protect us from getting the virus. If people experience some side effect, that is a normal sign that their body is building protection. Some bodies are more delicate and react strongly to the weakened virus injected into the body. Soreness and sizzling at the injection site, muscle ache, fever, chill, nausea, and tiredness are all common side effects of the COVID-19 vaccine. These side effects tend to be worse after the second shot. Several severe side effects from the vaccine are more common in the younger adult. So next is conclusion. In conclusion, not all vaccines currently being developed to prevent COVID-19 will be successful. Safety issue or a lack of protection will halt some. So a broad
portfolio of vaccine approach and technology progressing through human trial is reassuring. Ultimately, it is likely people need a repertoire of the COVID-19 vaccine to offer widespread protection. Different vaccine formulation will ensure vaccination is safe and effective for all members of society, including infants, the elderly, and people with a weakened immune system. So that's all from me. Thank you.